Hello, my name is Chao Lu, and I want to start by thanking the organizers for putting together this collaborative research network conference and giving us the opportunity to share our recent work on understanding the crosstalk between DNA and histone methylation, as well as its implication in developmental overgrowth syndromes. So we are a molecular and cell biology lab interested in understanding the role of histone and the DNA modifications in genome regulation. As many of you know, our genome is packaged extensively at several levels. At the basic level, 147 base pair of DNA is wrapped around octamer histone proteins known as nucleosomes. And from this structure, you can appreciate that for each histone, there are arm-structured tails extending from the nucleosome. And we know that both the histone tails as well as the DNA itself undergo extensive chemical modifications. So the DNA can undergo methylation and the 5-hydroxymethylation. And the histone tails, the lysine on them can undergo acetylation or methylation, and the serine can undergo phosphorylation, etc. So apart from the many cool biology about those epigenetic modifications, the enzymes that are catalyzing those histone and DNA modifications are often mutated in human developmental disorders and cancers, suggesting that those modifications have an important role in the disease development. For example, for today's talk, I will focus on Dmd3a, which, as you all know, is mutated in T.B.R.s and catalyzes the addition of DNA methylation. I will also talk about NSD1, shown on the right of the screen, which catalyzed specifically on the histone H3 lysine 36 dimethylation. And this is a gene that's mutated in another form of overgrowth syndrome, Sotos syndrome. So one intriguing feature of chromatin modifications and their associated enzyme is that there are various crosstalk mechanisms existing between them. So basically, any one modification on histone DNA can either positively or negatively influence the activity of another enzyme. And those crosstalk mechanisms really form the foundation of the epigenome domains, means that different histone and DNA modifications often coexist or display a mutually exclusive distribution patterns across the genome, forming those well-segregated epigenome domains. And those domains tend to be very stable and sometimes are heritable during mitotic division. And because the existence of those domains, it also allows the cells to achieve a very stable gene expression patterns. So as the cells undergoing differentiation, shown in this famous Waddington landscape, it is believed that those epigenetic domains are the basis for cell maintain their identity. So either the transdifferentiation or reprogramming becomes very challenging once the cell is committed to a specific lineage choice. So we're very interested to understand, are there new chromatin crosstalk? And what is the disease implication of those chromatin crosstalk? And for this, we were intrigued by human genetics data. So this is a figure adapted from a landmark study led by Dr. Tatton in 2017, where she and her colleagues have reported various mutations that underlie human overgrowth syndromes with intellectual disability. And among the known genes known for those overgrowth syndromes, a large part of them are related to epigenetic regulation. And we can see on this pie chart that various chromatin and DNA modifying enzymes such as NSD1, EZH2, and the Dmd3a, which is responsible for T.B.R.s, are all mutated in various forms of overgrowth syndromes. And the fact that all those epigenetic enzymes, when mutated in human, give rise to overlapping clinical features, to us, that suggests that at the molecular level, there are mechanisms for those enzymes to crosstalk, meaning that they perhaps are acting in a common pathway to regulate genes in a very intimate relationship. So to further understand this potential crosstalk between NSD1 and Dmd3a, we hypothesized that the product of NSD1, the H3k27 dimethylation, may play a role in either positively or negatively regulating the levels, recruitment, or the catalytic activity of Dmd3a, the enzymes for DNA methylation. So to do this, first we map the genome-wide distribution of various histone modifications and the Dmd3a. So shown in the top part of the screen is a chip sequencing study of H3k27 dimethylation, showing orange, which is the enzymatic product of NSD1. Also DMT3a, which is the enzyme that's mutated in T.B.R.S and catalyzes DNA methylation. We also provide a closely related modification on H3k36, so H3k36 trimethylation shown in green. And lastly, another de novo DNA methyltransferase Dmd3b shown in gray here. And I hope you can appreciate that we observe a very close co-localization pattern between H3k36 dimethylation and Dmd3a. On the other hand, Dmd3b tends to co-localize with H3k36 trimethylation, and this has been previously reported. We can repeat this analysis in a genome-wide fashion, and as shown here, we separated the genome computationally, sliced them into 10 kb bins, and for each bin, we calculate the amount of Dmd3a binding and the amount of Dmd3b binding, and correlate those bindings to the differences in the levels of H3k36 dimethylation and the trimethylation. So the more orange it is, means that that genome is predominantly occupied by K36 dimethylation, and green means trimethylation. And I hope you can appreciate that in the regions where Dmd3a binds, those are the regions that are dominated by H3k36 dimethylation. So next, we decided to go a step further and test if the activity of Dmd3a correlate with the levels of H3k36 dimethylation on a genome-wide level. And to do this, we take a data set from a 2015 paper. And in this data set, they also developed a very intriguing system where they use the DMT3 triple knockout mouse ES cells. So those cells have virtually 0 levels of DNA methylation. And they reintroduced either Dmd3a or Dmd3b into the cells. And they used the whole genome bisulfite sequencing to track where are the DNA methylation appear along the genome. So shown here, when we look at the genome and rank them by the levels of H3k36 trimethylation, that's shown on the left part of the panel, and we can see that the Dmd3b binding, but not the Dmd3a binding, correlates very well with the levels of H3k36 trimethylation. However, if we repeat analysis, but this time rank the genomic regions based on the levels of H3k36 dimethylation, we observe the opposite pattern, where the Dmd3a binding correlate, actually this is activity, correlates well with H3k36 dimethylation, and Dmd3b activity does not. So this is another evidence suggesting that there's a co-localization of not only the Dmd3a binding, but also its de novo DNA methyltransfer activity with this particular histone methylation mark, H3k36 dimethylation, that's catalyzed by NSD1. So as a next step, we wanted to perturb NSD1 and therefore deplete H3k36 dimethylation, and want to see whether it has any impact on either the binding or the activity of Dmd3a. So shown here on the left is again a chip sequencing snapshot of mouse ES cells, where shown on the top part are the parental cells, and the bottom part are the mouse ES cell, where we use CRISPR-Cas9 to delete NSD1. And you can appreciate that the levels of H3k36 dimethylation is indeed depleted upon the deletion of NSD1. And this depletion predominantly affects intergenic regions, where the genic H3k36 dimethylation were largely retained. Also importantly, the closely related mark H3k36 trimethylation remain largely unchanged when NSD1 is deleted, suggesting that it specifically affects H3k36 dimethylation. When we look at Dmd3a binding, we notice that correlating with the loss of H3k36 dimethylation, the intergenic binding of Dmd3a is largely lost, where Dmd3a actually redistribute into genic regions. So this is again supported by the genome-wide analysis shown on the right part, where we see a co-occurrence between the loss of binding of Dmd3a, the loss of H3k36 dimethylation, as well as the loss of DNA methylation. This is shown as in a heat map. So all three events are positively correlating with one another. And as I mentioned, it seems that NSD1 regulated H3k36 dimethylation mainly occur at intergenic regions. And we do see that if you classify the genome into exons, intron, and intergenic, loss of NSD1 specifically reduce the DNA methylation levels at intergenic regions. So mechanistically, we showed that the underlying basis for the preferential DMT3A recruitment by H3k27 dimethylation is through its PWWP domain. So this domain is common in both DMT3A and DMT3B and has been previously implicated in the interaction with H3k27 methylation. When we isolate the PWWP domain of DMT3A and we subject to the binding assay with a panel of recombinant nucleosomes carry different forms of histone methylation, as you can show here on the bottom left panel, we observe a very specific interaction of DMT3A PWWP domain with H3k36 methylated nucleosome, and specifically we observe a preferential binding of DMT3A PWWP domain with the H3k36 dimethylation modified nucleosome relative to the trimethylation. And this can be measured more quantitatively through the ITC assay in a collaboration with Haitao Li's lab. And for here, we subject, again, the PWWP domain of DMT3A to peptides that are either carrying no modification, mono, di, or trimethylation on H3k36. And again, we observe that they only interact with di and the trimethylation on H3k36, and the interaction is much tighter for the dimethylation compared to the trimethylation. We want to extend those both molecular and biochemical findings into patient samples. So as I mentioned, NSD1 is the defined a developmental syndrome known as Sotos syndrome. Previously, there was a Nature Communication paper that taking the blood samples from the Sotos patient, blood sample from the Sotos syndrome patients, compared that to the control samples, and the authors noticed a genome-wide downregulation of DNA methylation in the blood samples from Sotos patients. We're taking a step further, and we validated this finding, and indeed shown in this table here. If you look at the DNA methylation probes that are either up or down, compare NSD1 wild type versus NSD1 mutated samples, the Sotos syndrome have the majority of the probes are downregulated. NSD1 somatically is mutated in head and neck cancer, and we'll repeat the finding in the data set from the head and neck cancer. Again, if you look at NSD1 mutated samples, the majority of DNA methylation probes are downregulated compared to the few that are upregulated here. Moreover, when we look at the location of those probes that are downregulated, and we notice that there is a strong enrichment of the endogenic probes here. So shown here is the bar graph on the right that are, and you can see in the brown regions are the regions where probes are targeted to the endogenic region, and you can see that compared to all probes, the hypomethylated probes are enriched for those endogenic regions. And lastly, we took a look at the DMT3 mutation in T.B.R.S. So there are a couple of mutations that are directly affecting PWWP domain, and we have expressed the recombinant PWWP domain carrying those mutations and characterize them biochemically. And we found that all those mutations seems to be loss of function, meaning that they lost their ability to interacting not only the H3k36 methylated nucleosome, but just nucleosome in general. So we suspect that having those mutations, the DMT3A is not able to be recruited to the chromatin at all. And at the last analysis for this study, we compared the DNA methylation profiles from the T.B.R.S. syndrome patient and the Sotos patient that I talked before. And this analysis reviewed that indeed in T.B.R.S. syndrome and Sotos syndrome, their DNA methylation patterns are highly correlated here. So as you can show in this unbiased clustering, and if you look at the blue and the dark red columns, they're clustered closely together and away from all the control samples, as well as another form of overgrowth syndrome, Weaver syndrome patient samples. So taking all those data together, this recently published work, we think that there is a close crosstalk between DMT3A and NSD1, in that the NSD1 mediates H3k36 dimethylation, which is specifically recognized by DMT3A and facilitate its recruitment to endogenic region and maintain endogenic DNA methylation. In the setting of Sotos syndrome, there's a loss of NSD1, so the K36 dimethylation is lost. DMT3A is unable to be recruited to the endogenic region. And in the T.B.R.S., the DMT3A, the mutations really abolish its binding to chromatin, so the DMT3A is again not able to be recruited to the intergenic region. And a common feature between those two syndromes is that there is a decreased intergenic DNA methylation. So since the publication of this paper, we have been wondering what is the impact on gene regulation when you lose this intergenic DNA methylation and histone methylation landscape. So although there are not genes in those intergenic regions, those regions does harbor many cis regulatory elements that are important for gene regulation, such as enhancers. So we decided to specifically look at the enhancer landscape in cells that are losing NSD1 versus the control cells. So shown here, we performed the chip sequencing for H3k27 methylation, which is a mark for active enhancers in both parental cells, in the cells that are deleted for NSD1, or the cells that are expressing a mutation on histone H3k36. And we noticed that there is a group of enhancers that are losing H3k27 acetylation, meaning that they're becoming inactivated. This is shown in the bottom of this those histograms. And similarly, those enhancers lose H3K4 monomethylation. They also lose their chromatin accessibility as determined by ataxic. And when we look at the levels of H3K36 dimethylation and DM methylation, those tend to be the regions where we observe the largest decrease in H3K36 dimethylation and DM methylation, suggesting that those are regions that are affected the most when you lose NSD1. And also, interestingly, we observed that the levels of H3K27 methylation, so this is another histone methylation involved in gene regulation and particularly in gene silencing, they seem to be particularly increased at the region where they lose those enhancers. So this findings actually support nicely with what we previously known about those modifications, because both H3K36 dimethylation and DM methylation have an established role in inhibiting PRC2, which is the histone methyltransfer complex that specifically catalyzes H3K27 trimethylation. So you can imagine a scenario where normally NSD, at those active enhancer regions, NSD1 mediate H3K36 dimethylation will help establishing the DNA methylation, and both those two marks were inhibiting the activity of PRC2, so they're not able to lay down H3K27 methylation mark, and the genes are active. In the context of NSD1 loss, and then subsequent DNA methylation loss, PRC2 can now come in freely and putting on marks of H3K27 trimethylation, and these were inactive enhancers and leading to gene silencing. There is actually a small molecule inhibitor that are specific for PRC2 that's being approved to treat certain types of cancer. And we treated our cells with this inhibitor. And indeed, we noticed that in NSD1 knockout cells, the treatment of this particular PRC2 inhibitor is able to restore the enhancer activation landscape. So with that, I'd like to summarize the talk. I think overall, both our published study as well as our ongoing study suggests that there may be a unifying mechanism for the overgrowth syndromes that are associated with epigenetic enzyme mutations here. So I talk about the NSD1 and the DMT3A, and it's worth noting again from this graph from Dr. Tatton-Brown's paper, there are other histone-modifying enzymes that are mutated in various forms of overgrowth syndrome, including members of PRC2. So we think that a better understanding of the interplay between those epigenetic pathways will offer insight not only into disease mechanism, but potentially also give rise to novel therapeutic targets for those overgrowth syndromes, as well as in cancer where those mutations are somatically mutated. However, I think a big challenge, at least for us, is the context. We're mostly studying our mutations in the setting of mouse embryonic stem cells. And obviously, we need more disease-relevant models where we can both validate our findings and, more importantly, apply the same mechanistic principle, but to understand which set of specific genes are misregulated when those epigenetic pathways goes abnormal, and how that may translate into disease biology and a therapeutic approach. And with that, I'd like to acknowledge the folks that are involved in this work. This work would not be possible without the close collaboration with the three fantastic teams. So Dr. Jacek Majewski's lab and McGill, with Phil and Simone that are involved different in different parts of the story that I talk about are integral to our studies of the mechanisms of DMT3 recruitment and misrecruitment. Also, I'd like to thank Dan from Dr. Dave Ellis's lab in Rockefeller University. Dan is also has been spearheading all the studies, carrying on a tremendous amount of work to generate the mutation and profile those epigenomic distribution. And lastly, I think I mentioned Michael's name before, but Michael's team at EpiCypher has been instrumental in carrying out all the biochemical interactions between DMT3A versus chemically modified nucleosomes, which have given us critical insights into the underlying mechanism. On our end, I'd like to thank Sean in the lab, who's been helping with various studies, as well as the collaboration with Haitao Li's lab at Tsinghua University. And thank you for your attention, and I'm happy to take questions.